Bismillah ar-Rahman Rahim. Now we start the 18th chapter, and this is 5090 O-level syllabus, but this works for IGCSE and GCSE as well. This is the syllabus 2023, and we're going to do biotechnology and genetic modification, which is a slightly change in the new syllabus. So let's talk about this now. Now, as we go through the syllabus, biotechnology and genetic modification, the first 18.1 is about biotechnology and 18.2 is about genetic modification. Now, in biotechnology, we have to explain the role of the yeast in the production of bread and ethanol. Then understand that bacteria, so we need to know the structure of bacteria and why is it used in biotechnology and genetic modification. Why? Because they, number one, the only thing is they have a very rapid reproduction rate. They multiply every 20 minutes. And their ability to make complex molecules. So that's why we put the gene for insulin into bacteria, because they're able to make that insulin molecule. Discuss why bacteria are useful in biotechnology and genetic modification. Limited to, number one, there are no ethical concerns. So everybody is okay with, there's no religious issues, there are no ethical issues that, okay, well, we shouldn't use bacteria, just like there's animal rights and all that. So there are no ethical concerns over the manipulation and growth of bacteria. So we can do anything. We can manipulate them, we can grow them easily. Not a very nice thing, but we can. And then, of course, the presence of plasmids. So what are plasmids? We'll talk about that. Then we talk about how do we culture bacteria. So we use fermenters. And these are used for the large-scale production of useful products by bacteria and fungi, including the conditions that need to be controlled, limited to temperature, pH, oxygen, nutrient supply, and waste products. So we'll talk about the fermenters, these huge tanks where we grow these bacteria, and we can grow bacteria and we can grow fungi, like we grow the fungus penicillium, and it produces the antibiotic penicillin for us. Then number 5 is describe the uses of enzymes in biological washing powders. Why we use enzymes in biological washing powders is to remove the stains, protein stains. You spilled egg on your shirt. Now why would we have a washing powder? Because egg is albumin, protein. So in order to wash off that stain, we use biological washing powders. Then pectinase for fruit juice production. We want clear fruit. We don't want the particles of the lignin and pectin in it. So we don't want any of that. So we want a clear. So we use pectinase. And then we use lactase for lactose-free milk. But some people are allergic to lactose and they get bloating and they get problems. They get pain in their abdomen. So we have to get lactose-free milk, because lactose is a sugar which is found in milk. So all these are now in the 18.1, in which we're going to be discussing biotechnology. Now, coming to the first point, the role of yeast in baking bread and ethanol. And so the most important method, the traditional method in making bread was what? You added warm water, sugar, and yeast to a bowl, and you allow it to sit till it becomes bubbly, called proofing the yeast. You're just proving it's going to work. Then you add flour to the mixture to make a soft dough. So you've added the flour and the water and the sugar and the yeast, and now you're making a dough out of it. Now, knead in the flour. Knead means you sort of press into the flour and smooth and become smooth and elastic. Allow to rise fermentation until it is doubled in size. So if you had that in a beaker, this was the dough. Now dough is going to rise, double its size. Then you punch down, divide and shape and allow to rise again, and then you bake. So this was the traditional method of making bread. Now, when you look at it, what is fermentation? Fermentation, also known as alcohol fermentation, is the breakdown of sugars by bacteria and yeasts using a method of respiration without the need for oxygen. These bacteria and yeast are microorganisms, and what do they use? They use glucose, fructose, and sucrose are converted into energy for these cells. The products of this process are ethanol and carbon dioxide as metabolic waste products. When the solution reaches about 15% ethanol, then the enzymes can't function and the process of fermentation ends. Why? Because ethanol is toxic. It is going to kill the yeast and the bacteria, if any present. Now, looking at the equation for fermentation is very basic. You see, you've got to understand that glucose is respired anaerobically. That's why it's called fermentation. It's another name for anaerobic respiration. We're going to produce ethanol and carbon dioxide. Now, in bread, we need the carbon dioxide to make it rise. In the production of ethanol, it's just the ethanol that we need. So ethanol and carbon dioxide are two main products, and we're using the microorganism is yeast. Fermentation is used for bread making and the production of wine. It is also used to make products such as cheese, yogurt, and soya sauce. The word equation is glucose ethanol, and the symbol equation is this. So C6H12O6 gives you 2C2H5OH plus 2CO2. Now, the second point of the syllabus is why bacteria are used in biotechnology and genetic modification. See, they're two separate things. Biotechnology is this bread and wines and in brewing, but genetic modification is when you change the genetic makeup of an organism somehow. We're going to talk about it how. Like we take the gene for insulin from a human being and put it in the plasmid and then put the plasmid back into bacteria. Now we've got genetically engineered, genetically modified bacteria, and these will produce insulin for us. But in biotechnology, we are just going to use, like bread making is biotechnology, but genetically modifying bacteria producing insulin is genetic modification. Okay, now why do we use bacteria? The first question is that number one, they have a rapid reproduction rate. Number two, they can make complex molecules for us, like they can make insulin for us. We need insulin because people who are suffering from diabetes need the insulin, but we can actually put the recipe for making insulin into a bacteria, and the bacteria will make insulin for us. So they can make complex molecules. Now, there's no ethical concerns. No ethical concerns over manipulation of growth. You see, as I'll talk to you later, when we used other animal products, then we had problems. There were religious ethical issues. There were other issues that people who are vegetarians would not take any animal products. And the fourth, now these are 4 points which everybody must remember. Rapid reproduction rate. Number two, makes complex molecules. Number three, no ethical concerns. Bacteria is not haram or halal, or it is not banned in certain religions, or it is allowed in certain religions, or certain people say, okay, well, we can't have bacteria because you take yogurt, yogurt has bacteria in it. Then the fourth and the most important point is that there's the presence of these circular uh, little circles of DNA, which are called plasmids. Now, these are the ones which are very important, which we can actually modify and then put them back into the bacteria. So these are the four important things: rapid reproduction rate, make complex molecules, no ethical concerns, and presence of plasmids. Now let's discuss the first point, rapid reproduction rate. Now, how do bacteria divide? They divide by binary fission. Please remember, this is not mitosis. This is not mitosis. Everybody should be very clear. This is not mitosis. Binary fission is a totally different method. In mitosis, there are certain stages of mitosis like prophase, metaphase, anaphase, and telophase. Well, we don't discuss that in O levels, but we do talk about it just a little bit. Now, this is binary fission. Now, what happens in binary fission? Cell replicates its DNA. First, the DNA. This was the original DNA. Now, a copy of that DNA has been made. So, cell replicates its DNA. There is a cell wall in bacteria, there's a cytoplasmic membrane, there's a replicated DNA. Now, number two, the cytoplasmic membrane elongates, separating the DNA molecule. So now the cell has become a little longer. Then the cross walls form, membranes invaginates. Cross wall form completely, and two daughter cells formed. Now this happens every 20 minutes. So now imagine that there's a place, so there's some beaker in which we have 1 million bacteria. In 20 minutes, there are 2 million. And then the 2 million are 4 million. So every 20 minutes, they're going to become double. So 1 million, 2 million, 2 to 4, 4 to 8. So you realize that this is enormous. This is a very, very rapid reproduction rate. So when you genetically modify bacteria, you can just grow them in fermenters and provide them the nutrients, and they go to multiply every 20 minutes. The second point is why do we use bacteria is that they make complex molecules. Whether we use bacteria in biotechnology or genetic modification, the reason is that they make complex molecules. They can make complex molecules. Now, for instance, I said Let's take the gene for insulin and put it in bacteria, and the bacteria can make insulin. Now, what you have to realize is that insulin is a hormone, it's a protein. So for protein synthesis, you need only two things. Number one, you need the DNA. Now, how are we going to? So now we've got to understand in bacteria, there's two types of DNA. One is this nucleoid DNA, which is this one, all this, right? And besides these are little circles of DNA which are called plasmids. This is a plasmid, right? So what we do is we take out this plasmid and we change it. How do we change it? We cut it and then we add another enzyme, another portion of DNA from a human cell or from any other organism, whichever we want. And then we put this back into the bacteria. So now we've got this DNA, which is now alien DNA. And then what is going to happen is this DNA is going to result on the ribosomes, because please remember that all bacteria have ribosomes. On the ribosomes, the protein molecule is going to be made, because DNA is the recipe to make that protein. Say this DNA is for insulin. Now then the amino acids are going to be used, and on the ribosomes the insulin will be made. Why the ribosomes? Ribosomes is a site of protein synthesis, where what is done is that the amino acids are assembled together to make a protein molecule. So what you have to understand is that they can make complex molecules. So that ability is being, then we are exploiting that. We are in fact manipulating the bacteria so that it can now do things for us which we want it to do. The next part is that biotechnology and genetic modification. Why do we use bacteria? Is because there are no ethical concerns. Now, for that, I have to go back and tell you about how we used to get insulin from the pancreas taken from animals, and then the insulin was extracted. But there were religious concerns, because sometimes we used cow's pancreas, sometimes we used pig's pancreas. Now, religiously, some people are not okay with cows, and some people are not okay with pig pancreas. So with bacteria, there are no such issues. And then, of course, as I told you, some people are vegetarians. They do not use any animal products. So they would not have used this insulin either. So this insulin, which was extracted from animals, had a lot of issues. We'd had to slaughter a lot of animals to get their pancreas. So a lot of animals were, unnecessarily killed because we wanted their pancreas. Then, as I said, religious issues, and as I said, some people vegetarian, so they would have issues. But in bacteria, there were no such ethical concerns. Now, another very important point was that bacteria have plasmids. So we're saying presence of plasmids. You see, other organisms do not have plasmids. A human cell does not have plasmids. So bacteria had plasmids. Now, what are plasmids? They're little circles of DNA, as I can show you here. So what we do is we take out this plasmid, as I've shown you by this arrow. We've taken out this plasmid, which was inside this one here. We've taken it out. And now what have we done? First, we cut it. So this plasmid was like this, and then we cut it, and then we add. Now this is a human chromosome, which I have just drawn here. So this was the human chromosome. And now I have removed this part. And so this part now is, this is the part here which was removed. Now this part I put here, and then I've used an enzyme to join it. Now this is called the recombinant DNA. Recombinant DNA is a new combination of DNA, because some of it is bacterial DNA and some of it is human DNA. And now this recombinant DNA we are putting back into a new bacteria. We got it out of this one. Now we're putting out in a new bacteria. And now this bacteria we're going to culture in a fermenter. And then this bacteria is going to produce insulin for us, because we put in a gene for insulin taken out of a human chromosome, into a plasmid, and then we have put this recombinant DNA back into the bacteria, into a new bacteria, not from this one. This one we have destroyed, because when we actually destroy it, we actually break the cell wall and the cell membrane to extract these plasmids. And then we put it. So the presence of plasmids is a very good tool for us. For genetic engineers, plasmid is a very good tool to take it out, then genetically modify it, and then put it into another set of bacteria, and then culture those bacteria, and those bacteria will produce that protein for us, for which we have put the gene. Now we're going to discuss what is a fermenter. Now a fermenter is basically a big tank which is used to culture bacteria or microorganism, even bacteria, even fungi. Now, what does it basically have? Let's look at the first one, cooling jacket. Why do we need a cooling jacket? If there are lots of people in one room, it becomes very hot, it becomes very stuffy, because everybody's at 37 degrees Celsius. So similarly, in a fermenter, we have millions and millions of bacteria. They are all respiring, and energy is being released. I said energy is being released, not energy is being produced. Energy is being released, so it becomes hot. So you need a cooling jacket to keep the temperature down. Now this is an impeller. I'm just going to give you the definitions later on. Then there's a temperature probe. Then there's a pH probe. Then this is the culture medium, which is the nutrient solution which we have put in for them to actually multiply. Then this is a top plate. Then this is a stirrer. Then this is an acid base, pH sensor. Then anti-foam, inoculation pipe, baffle, oxygen sensor, sampling point, and compressed air inlet, and then sterile air line, right? Now, if you look at the another explanation of all this, what is the first one? The impeller. It's an agitator to stir the medium continuously, hence prevent cells from settling down and distribute oxygen throughout the medium. Impeller speed decreases as the size of the fermenter increases. Sparger or an aerator introduces sterile oxygen to the media in case of aerobic fermentation process. Baffles disrupt vortex and provide better mixing. Inlet air filter. Filter air before it enters the fermenters, because in the air there might be other bacteria or fungi or fungal spores. We don't want those to grow. We only want a certain specific type of bacteria which we have put into the fermenter to grow. Then a rotometer measures flow rate of air or liquid. Then a pressure gauge measures pressure inside the fermenter. Then there's a temperature probe, measure and monitor change in temperature of the medium during the process. Then there is a cooling jacket to maintain the temperature of the medium throughout the process. Then the pH probe measures and monitors pH of the medium. Then there's a dissolved oxygen probe, measures dissolved oxygen in the fermenter. Then there's a level probe, measures the level of the medium. Then there's a foam probe, detects the presence of the foam. Then a sampling point to obtain samples during the process to check whether there's any contamination or not, or should we just stop it and then we have to clean up the whole fermenter and then start it all again. Then there are valves which regulate and control the flow of liquids and gases. Another very simple basic design. I like this diagram very much. It's a product outlet, stirring paddles, exhaust outlet, aerator, motor, acid base inlet, nutrient inlet, and a sensor probe for pH, O2, oxygen, heat, etc., and a water jacket. The next thing we want to discuss is biological washing powders. Now, the normal washing powders that we have are not written. You have to really see it, whether you want to buy a biological washing powder or another non-biological washing powder. Now, the majority of commercial enzyme production involves protein digesting enzymes, proteases, and fat digesting enzyme lipases for use in the food and textile industry. When combined in washing powders, they are effective in removing stains in clothes. Now, for instance, you're eating your omelette and you've dropped some egg on your clothes. Well, that is going to cause that's a protein. Or, like in operation theaters, when we remove the operation is done, all that which was used, the gowns or the doctors and the other things that were used on the patient, they have nothing but blood stains on them. And blood is all mostly protein, plasma proteins and proteins of cells. So In order to remove these stains of protein, blood, gravy, fats like grease, you dropped oil on it or you drop some butter on your clothes. So these protein and fat molecules tend to be large and insoluble, but when they've been digested, the products are small soluble molecules which can pass out of the cloth. So biological washing powders save energy because they can be used to wash clothes at lower temperatures, and there's no need to use boiling water which will wash off the stains. However, if they are put in water at high temperature, the enzymes become denatured and they lose their effectiveness. So biological washing powders need to be used in warm water, not hot water, because the hot water is going to denature the enzymes and I basically be using the enzyme to wash off the stains. But if we're going to use hot water, well, the enzyme will be denatured and it won't really work. Now, the next topic is investigating the use of pectinase. Pectinase is an enzyme which actually dissolves the solid particles in the fruit. So when you crush the fruit or you're drinking out the fruit juice, what you do is if you add this pectinase, then more of the juice is extracted. So it says investigating the use of pectinase in fruit juice production. Now, make 100 centimeter cube of apple puree using a liquidizer or use a tin of apple puree. Apple puree, you understand, you just take the apple and just crush it. Put it without any water or anything, put it in the blender and just get the puree out of it. Transfer the puree to a 250 centimeter beaker. Add 1 level teaspoon of powdered pectinase enzyme and stir the mixture and leave it for about 5 minutes. Place the funnel in the top of a 100 centimeter measuring cylinder and line the funnel with a folded filter paper. Transfer the puree into the filter paper. So you've got the funnel, right? And you've got the filter paper. So place a funnel in the top of a measuring cylinder and line the funnel with a folded filter paper. Now you transfer the puree. The puree is transferred here into the filter funnel and leave it in a warm place for about 24 hours. But you had added pectinase to it. Remember that you transfer this 100 centimeter apple puree, then you added 1 level teaspoon of powdered pectinase, then you placed it, then you transfer the puree and leave it in a warm place for about 24 hours. Other measuring cylinders could be set up in the same way with puree left to stand at different temperatures to compare the success of juice extraction. Now we're going to be really looking up is okay, how much juice did we get out of this apple puree? Did we get 50 cm cube, 60 cm cube, 80 cm cube? Because what is it going to do? It is going to crush the cell walls and the cell membranes and everything. The pectin is going to remove all that so that the juice from inside can all come out. So it is the use of pectinase in fruit juice production. Please understand that. We also use pectinase in clarifying when we get very clear juices. When we want to have clear juices, even then we want to remove the solid particles in it. So we also use that in that process. But here it is not about that. It is about the fruit juice production. Now, the last topic that we are going to do is lactose-free milk. Now, why do we need lactose-free milk? Because some people suffer from lactose intolerance, which is a digestive problem where the body does not produce enough of the enzyme lactase. As a result, the lactose remain in the gut where it is fermented by bacteria, causing symptoms such as flatulence, wind, diarrhea, stomach pains. Many foods contain dairy products, so people with lactose intolerance cannot eat them or suffer the symptoms described above. However, lactose-free milk is now produced using the enzyme lactase. A simple way to make lactose-free milk is to add lactase to milk. The enzyme breaks down lactose sugar into two monosaccharide sugars, glucose and galactose. Both can be absorbed by the intestine. An alternative large-scale method is to immobilize lactase on the surface of beads. The milk is then passed over the beads and the lactose sugar is effectively removed. This method avoids having the enzyme molecule in the milk because they remain on the beads. The food industry uses lactase in the production of milk products such as yogurt. It speeds up the process and makes the yogurt taste sweeter. Because you see, the lactose is less sweet, so when it's broken up into glucose and galactose, then it makes it more sweet. There'll be more glucose, more sweet naturally, because glucose is very, very sweet. So lactose-free milk is produced for people who have lactose intolerance. This is people who suffer from lactose intolerance. So they cannot have any dairy products. They cannot have any cakes and pastries which have cream on it, and they will have problems. So we can produce lactose-free milk in this manner. This completes 18.1, and this is the first video on chapter 18. One more video on this, and that should complete this chapter, inshallah.